Welcome everyone to the first webinar of a series of webinars of the CompBioMed project. This series of webinars is endorsed by the Virtual Physiological Human Institute. My name is Britt van Rooij. I'm a member of the VPH Institute PhD Student Committee on behalf of CompBioMed. CompBioMed is a user-driven center of excellence in computational biomedicine to promote an uptake, exploitation of high-performance computing within the biomedical modeling community. And the users' communities come from areas in different areas, so academia, industry, and clinical practice. So the title of today's webinar is HPC Simulations of Cardiac Electrophysiology using patient-specific models of the heart. A special welcome to our speakers, Anna Micholet and Francesc Levrero, both from the Computational Cardiovascular Science group of the University of Oxford. They will present their insights of their latest results on high-performance computational simulation of cardiac electrophysiology using patient-specific models of the whole heart. The webinar will focus on the well-known simulation software CHASTE and Alya. So Anna is a senior researcher in the Computational Cardiovascular Science group. She received her PhD in biomedical engineering from the University of Zaragoza in 2011. In 2012, she joined the Oxford Cardiovascular Science group. Her current research interest includes electrophysiology modeling and simulations and biomedical signal and image analysis with the aim to understand mechanics and provide biomarkers for cardiac diseases, making use of in silico simulation and patient-specific data. Francesc is a research associate in computational cardiovascular science. He finished his PhD in civil engineering at the University of Edinburgh in the topic of multi-scale solid mechanics and HPC applied to trabecular bone. In 2016, he joined the CCS group as part of the CompBioMed project, and he's in close contact with the Barcelona Supercomputing Center. His topic is the study of multi-scale behavior of the human heart under healthy and diseased conditions. So before we start, I'd like to mention if you have any question, you can type them in the question tab, so in the webinar window. And now I'd like to give the floor to our speakers.
Hello, good morning. First of all, thanks a lot for registering to this CompBioMed webinar. My name is Anna, and this is the outline of the webinar today. We will start with the introduction of heart health electrophysiology. Then we will follow with mathematical modeling, and then we will introduce both softwares of today, CHASTE and Alya. And finally, we will finish this webinar with the final comments. Well, this is a brief introduction of electrophysiology. The heart is a contracting muscle. It has 4 chambers, 2 atria and 2 ventricles. Those 4 chambers, the right atrium receives blood from the body that it's pouring oxygen and pumps it to the right ventricle that it's here. From the right ventricle, the blood is pumped to the lungs and comes to the left atria that receives blood rich in oxygen from the lungs and pumps it to the left ventricle. And from here, the left ventricle pumps the oxygen-rich blood to the body. Then we have 2 systems. One is pulmonary, that occurs between the heart and the lungs, and the other is systemic, and occurs between the heart and the body. We have talked about the mechanical process, but this mechanical process is triggered by electrical activation that I will show you in the next slide. Everything starts in the sinoatrial node. This is the natural pacemaker of the heart and excites the right atria and then the left atria. From some pathways in the right atria, it gets to the atrioventricular nodes, where through the bundle of His propagates very fast through the septum and then from the bundles to the right and left ventricle. That leads to the Purkinje fibers that allow propagation throughout all the endocardium. Then there is a cell-to-cell propagation through gap junctions and stimulate all the heart. So this is a multi-scale process. As you can see, different regions in the heart presents different electrical properties. Here we have action potential. The action potential is the electrical activity of the cell between inside and outside the membrane. It has an activation phase, and then it has a repolarization phase. Heterogeneities in activation and repolarization are also reflected at whole organ level, at body surface level, as the electrocardiogram. The electrocardiogram is the electrical activity that is measured at the body, at the torso level, by placing some electrodes in our torso. So as I said, this is a multi-scale process. It goes from cell to whole heart, then it propagates to the torso, and from here, if we place some electrodes, we're able to calculate the 12-lead ECGs. We start with electrophysiology. As I said, it's called action potential. It has 4 phases. The first one is the upstroke. Then it has a notch. This is the phase 1. Then it has a plateau here. That is the phase 2. Then repolarization. And then the phase 4 is the resting potential. There are ions that travels through the membrane, and they are present in different phases of this action potential. In particular, sodium and calcium currents are involved during the first and second phase of the action potentials. And potassium-based currents are involved in the repolarization phase. Also, other currents like IK1 are involved in the resting, in the values of the resting potential. All these cells are connected by junctions, gap junctions, that form what we understand as fiber, cardiac fibers. Through these gap junctions, the cells diffuse easily from cell to cell. So once they are organized by fibers, the electrical activity propagates much faster through the fiber direction and slower perpendicular to it. So as I said, all these electrical properties that goes from ionic current, single cell, tissue, organ, and body, they can be modeled mathematically, that we will explain afterwards. In our group, we model from ionic current to single cell, tissue, organ, and body. And the idea is that these ionic currents models, they are included in our cell models. They are organized into cardiac fibers that belong to a specific heart geometry, and this heart can be embedded in a torso. When we simulate the electrical activity through all these scales, we're able to have the body surface potentials, and from there the ECGs. This is a multi-scale process, so it's computationally very expensive, and then single cell and 2D models, they can be run in a PC, while bigger simulations, they need clusters to be run. And now we will start with the mathematical model. Francesc.
Hello, everyone. First of all, thanks for registering again. And my part that I'll give before we go to the explanation of the different software packages we consider is regarding mathematical modeling. So we'll start by a brief explanation of what a cell AP model is. AP stands for action potential. The first one for cardiac cells was developed by Dennis Noble, who is or was probably still is an academic in Oxford. This is the shape of the action potential. You can see it here, and this is a schematic representation of the model with all the currents that they're considered in this. So he developed this cardiac cell model based on Hodgkin and Huxley formulations, and even some of the most modern models still use these sort of formulations. We'll see it later. So what do we consider as let's say, gold standard cell model? What we consider is the O'Hara-Rudy model. Its schematic representation is here. Why do we do it? Basically because it is based on more than 150 human hearts, experiments on these hearts. Some of the ionic hearts are still based on the Hodgkin and Huxley formulation, which we can see here. And this is a schematic representation of an ion channel. What Anna said is that the cardiac electrophysiology problem is a multi-scale problem. Why did she say that? So here we have maybe the two most employed models of diffusion, electrical diffusion in cardiac tissue. This is the monodomain and this is the bidomain. The difference between them is that the bidomain considers different diffusivities for the intracellular domain, so inside of the cell, and extracellular domain outside of the cell, while the monodomain, what it considers is that these two diffusivities are proportional, so they have basically the same directions, and there's only a constant of proportionality which differs. You retrieve the monodomain model by basically putting this here and then substituting back to the diffusion equation. This is what we see here, basically. When we put all these models together, what we retrieve is this, in this case, the monodomain, but in a way, each term represents something different. This is the diffusivity of the tissue, which is represented by this Laplacian here. These ion currents come from the cellular model and are all of these sodium and calcium currents that we saw before. The I-stim is the term that I'd like to define here. This is a stimulus current that we use to trigger the problem. So basically, if we don't use a term like this, or if it's zero throughout the simulation, the model is completely stationary. And when you put a value which is high enough, then it will trigger a pulse, which will propagate through the heart, through the cardiac tissue that we are considering. And now I'd like to give a few data, a few characteristics of the software we're considering. First is CHASTE, which is developed at the University of Oxford. And then Alya, which is, here it stands for High Performance Computational Mechanics, but it's actually multi-physics. And this one is developed at the Barcelona Supercomputing Center. So basically, you can run with both any spatial dimension, ZOD being a cell model, so a system of ODEs, basically. CHASTE uses PETST as the linear solver and METIS as the domain decomposition method for parallel clusters, basically, while Alya uses in-house linear solvers and METIS as well. CHASTE is written in C++ and Alya in modern Fortran, and by modern, I mean 95 plus if you want. To solve the cell action potential models, CHASTE uses different methods for Euler, backward Euler, Runge-Kutta, or any method which is included in the set in the software CVOD, while Alya at the moment only uses forward Euler. CHASTE is multi-scale, as we said before, and Alya is multi-scale and also, in a sense, multi-physics. You can also calculate CFD if you want, for instance. Both are highly scalable, but I'd like to mention that Alya, the latest test we've seen, mentioned that it scales up to 100,000 cores on NCSA's Blue Waters in the States, the supercomputer. So now I'd like to give the word back to Anna, who will explain a bit more about CHASTE.
Okay, I will start by talking a little bit more about the cardiac CHASTE functionalities. CHASTE is a multi-scale simulation package, and its name stands for Cancer, Heart, and Soft Tissue Environment. It's a general purpose simulation package, and it's aimed at multi-scale computational demanding problems arising in biology and physiology. CHASTE includes several modules. One of them, that is the one that I'm going to talk about, and the one that I'm a user, is the cardiac CHASTE. And these are some of the cardiac CHASTE functionalities. It's able to solve monodomain and also bidomain models. They have an automatic implementation of cellular action potential models from the CellML repository, where you can find all the cells models in this format, CellML. It has the automatic generation of mathematical models for fiber orientation, stricter, whatever. And then it's able to do checkpoint of simulations midway through run and restart with altered parameters, and allows some functionalities for post-processing and also output results of the simulations. And it can calculate electrophysiological properties such as action potential duration maps, activation time maps, also conduction velocity. Well, you have a lot of information in this web page and plenty of tutorials, and it's very well explained. In this webinar, we plan to show you an example of a 2D electrical propagation using CHASTE. In this case, we have used a 2D mesh geometry coming from magnetic resonance image. Here we have the left ventricle, here we have the right ventricle, and from here we were able to develop this 2D mesh. We will start with a very simple example of a controlled 2D electrical propagation in which we will include a stimulation site in the septum. The properties of this simulation is that it's a 2D tissue from MRI, it has this mesh. We will use the O'Hare-Rudy 2011 epicardial model. We are not going to include in this example heterogeneity. So in the whole heart, all the cells will behave like this. Then we are using bidomain model for propagation. We have isotropic conductivities, and we will apply a regular stimulus in this site of 600 milliseconds of period. So yes, with these 60 lines of code allows to simulate the electrical activity with the O'Hare-Rudy model and bidomain in CHASTE. So we can divide the test, this code, in 3 parts. The first block includes the library. Which are the libraries that are included? We have here the test suite to create our CHASTE test. We have also included PETSI, that is the library for scientific computing, and is needed to run cardiac simulations. Then we're going to add the bidomain problem class, as we said, that is we are going to solve the bidomain model. Then the class for regular stimulus, we're going to apply a regular stimulus of 600 milliseconds. And then in order to generate and read meshes, tetrahedral and also triangular meshes, we're going to include this class. And finally, the O'Hare-Rudy model, specifying that we're going to use the numerical solver CBOD, that is an adaptive numerical solver. Sorry. The second block declares a class, that is the cell factory, that is needed for cardiac simulations. We define the cell properties for each of the nodes of the 2D geometry. The cell factory tells the problem class what time of cardiac cells to create. So for each of the nodes, the coordinates x and y are extracted, and if those coordinates belong to the stimulation site that is in the septum, as I showed you before, then we define a point. I will start here. We define a pointer that is pcell, that has the characteristics of the O'Hare-Rudy model and also specifies the way of solving this. So Then we extract the coordinates of that specific node, and if it belongs to the stimulation region, then we define this stimulus. Otherwise, the cell won't have any stimulus that is defined as this one. And here we have the third block, that is the principal class, where the problem is specified and inherits the application. We define the problem. We first specify the file path where the mesh is located, and then we read that mesh. Then we declare the variables regarding the ODE time steps and the PDE time steps and the printing time. The ODE is used for solving the cell models. The PDE is for solving the bidomain PDEs. And the printing time tells you how often the output is written to file. We have defined these characteristics. And then we set those characteristics. Then we have the bidomain parameters, in which we specify or we set the surface area to volume ratio, also the capacitance, and then we specify the intracellular and extracellular conductivity, since we are using a bidomain model. Normally, we have here two numbers. Normally, if not fiber orientation are specified, that is our case, the default longitudinal, that is the first number, and perpendicular directions are the Cartesian. So this is X and Y. As we said before, it's going to be an isotropic propagation. And then we specify the simulation time, that it's 1,200 milliseconds. Then, as we said before, CHASTE provides with functionalities allowing post-processing and also output results. We have made use of, in this example, of the functionality of getting an APD map, axion potential and duration map, in which for each of the nodes of our mesh, we will have the duration of the axion potentials. Then we have used also the activation time maps that tells us when the cell has been activated, then the maximum upstroke velocity map, and then we have some output options that tells you which is the directory in which the results are going to be stored, and also the name, the prefix name, and also we are specifying that the output results are going to use the original node ordering as the mesh that we use as an input. Then we said that we don't want to save our results in this format, the Meshalizer program that opens that format, and we are saving in parallel VTK. We have to say that we have saved also an H5 format, the results in its format. And then we specify the period of the regular stimulus, that is going to be 600 milliseconds. Then we make an instance to the cell factory here with the period of the stimulus, that is 600. And finally, we define the problem as a bidomain problem with the previously defined cell factory. And then we initialize and we solve it. And this is the results that we obtained by running this test. We have here the APD map. As we are expecting, we have similar values, APD values in all the geometry. Then the activation time map that starts from the septum, and then it propagates. And then similar maximum upstroke velocities all over the tissue. And here, of course, we have showed the results that are the voltage that is now activated, and this is millivolts, 40 millivolts, and now it's repolarizing. Now we are going to increase the difficulty. We're going to include in our example a region of ischemia. Ischemia in the anterior region. As you know, electrophysiological changes are arising from ischemia. The main ones are hyperkalemia, that is an increased extracellular potassium concentration. Normally, under control conditions, we have about 5 millimolars, but we are going to set it to 8.5 millimolars. Then hypoxia, that is a reduction of oxygen, that is normally modeled by activating FK-ATP channels 4%, and acidosis, that is modeled as a decrease of the sodium and L-type calcium currents. We're going to decrease it 25%. Here we have an example of what we are expected to observe. Here we have an action potential of a control in blue and an action potential of an ischemic cell in red. The hyperkalemia increases the resting potential, the hypoxia decreases the action potential duration, and acidosis decreases the amplitude and also decreases the slope of the upstroke. And the O'Hara-Rudy model doesn't have those characteristics, or we had to include the FK-ATP channels. So here there is a reference to that tells us the model that we have used that is based on the O'Hara-Rudy model. So the main differences are the following. We have declared as attributes the three components of the ischemia as members of the cell factory class, and they are also specified in the constructor. And then we have included also this line here. It was if the node is in the stimulus region to this, if it's in this ischemic region, then we will set the extracellular potassium concentration to the new value. We will get the parameters of the fast sodium current conductance and also the L-type calcium current conductance, and we multiply them by the scaling factor that we define in the constructor. And finally, we will set the ATP-dependent potassium current conductance to the value that we have telling the problem. And of course, we have specified those parameters in the definition of the problem in the third block. Okay, and these are the results. We can observe here that we have shorter APD values as expected in all the ischemic region. We have also the activation time map. We cannot see that normally in the ischemic region, it's the activation time maps the conduction velocity gets slower, but we can see it here. Here we see that the maximum upstroke velocities are slower in the ischemic region. And here we have the voltage. Now the cell is depolarized, and now it's repolarizing. Okay, and we are going to complicate it a little bit more, that it's just like including also an ectopic activity close to the border zone of the ischemic region. So in order to do this, the only thing that we have done is like including as a class the simple stimulus, and also in the cell factory defining it, and this is the amplitude of the stimulus, the duration of the stimulus, and the time in which you have to apply that stimulus. And also you define the ectopic region, that it's an ellipsoid in which you define the center, you define the radius, and if the cell is in that region, then we will apply this stimulus at the specific time. And this is the results. Now it's depolarized. Now it's repolarizing. The ectopic stimulus is in 250 milliseconds. So we had a conduction block in the ischemic area, and this geometry facilitates a re-entry. We observe like it's coming back, and it's not repolarizing. Okay. Well, and we can complicate things more. We're not going to show examples now about the CHASTE codes, but we are going to show the results of 3D simulations in CHASTE. Here we have a biventricular mesh in which we have specified an ischemic area, but here in this case we have also specified our border zone area next to the epicardium, but also in the endocardium. And also we have complicated this a little bit more by including the physiological transmural heterogeneities from epicardium having a shorter action potentials to endocardium having a longer action potentials. And in this specific work, we were also applying an ectopic activity close to the border zone. You will see it here. And conduction block and re-entry. And we are seeing how it looks like this arrhythmia. The good thing of multi-scale simulation and mathematical models is that if you're interested in a particular area and a particular time frame, you can choose it and you can see what is happening at cell level. And you observe that there is some abnormalities at cell level. And now our group is also moving to personalization of MRIs. Oops, I think it hasn't moved. Sorry, could you see my screen?
Yes, we can.
Okay, now?
Yes.
It's the new one, okay. That's working. So we are moving, we have a research line that is personalization of anatomical models. We start from MRI acquisitions. We use localizer that are some images that the clinical doctors uses to locate the heart, and we segment the torso of from those images. Also, we use the Cine MRI, in which we have the 4 chambers view and short axis view, and we have segmentations in order to construct the heart. So from here we construct the heart, from the localizer we construct, and together with a statistical model we construct the surface of the torso. So finally we end up with this kind of geometrical models of personalized to each of the patients. Once we have this, we need to parameterize with an action potential model, as we said, and all the heterogeneity that you want to include in your geometry. Also, we include fiber orientations. As I said before, the electrical activity goes faster along the fiber direction and slower perpendicular to it. And then we have to set, or we have to set an activation model to simulate Purkinje, and then we're able to simulate the electrical activity with all these ingredients from ionic level to whole organ and then to whole body. And by, of course, placing some electrodes in the body, we're able to simulate the 12-lead electrocardiogram. And this is one of the examples that we have, and here we have the 12-lead ECGs, and now it's repolarizing. Okay. And just the last slide to say that computer simulations can explain cardiac phenotypes, and we can use this kind of multi-scale simulations to be able to extract some information from these cardiac phenotypes at ECG level. And then we go to Alya.
Hello, this is Francesc again, so I'll be explaining a little bit more about Alya. So these are additional functionalities that I didn't mention before. We have monodomain and bidomain models available for the tissue propagation. In terms of action potential models, cellular action potential models, we don't have that huge variety of CHASTE because we don't have this implementation of CellML, but we do have a few quite relevant ones, Fitsunagumo, Hararudi, and Tentusher, and at the moment they are hard-coded, but we're working on an easy interface to be able to easily implement new ones. The bidirectional electromechanical coupling is implemented and fully functional, and by bidirectional, I mean from electrical to mechanical and from mechanical to electrical, and I will explain a little bit more about this later. Excitation contraction coupling models and stretch-activated ion channels are also implemented, and this also comes when I mentioned the bidirectionality of the coupling. Alya is highly efficient and highly scalable, as I mentioned before, like tests on like hundreds of thousands of cores, 100,000 cores have been already performed on American supercomputers, and the scalability is almost linear, so quite impressive. And you can use other physics, so you're not limited to only electromechanics. You can use combustion, like CFD, even chemical diffusion, and so on. This is an example that I will show the input files later. It's a biventricular model, 5 million nodes and 32 million elements. It's run on Mare Nostrum 4 on 2000 cores, and it runs in 15 minutes, but I'd like to mention here that around 10-12 minutes of that time is actually spent on pre-processing, so the actual running is really just a few minutes, so it's really, really fast. The example that I'm going to show contains 4 input files in ASCII format, which quite a straightforward format, and we'll see it later. And this example is just electrical propagation, and we'll see it here. We start from the root nodes that simulate where the Purkinje fiber, when the bundle of his actually meets the endocardium, and then there is a fast, I'll run it again, there is a fast propagation through the endocardium, and then it diffuses through the rest of cardiac tissue. Now it's repolarizing, and there we go. Again, just in case, because the internet connection seems to be playing some games now. Yeah. And now it's repolarizing. Yeah, okay. I'll show a small example later of a small electromechanical simulation, and don't get too scared by these equations. It's just that I want to explain what this bidirectionality of the coupling means. So this is our solid mechanics problem, equations of motion, and the corresponding stress-strain constitutive law. This is the Holzapfel and Ogden model, an anisotropic model. This is an active contraction model, what I mentioned before as excitation contraction model. It's a set of ODEs that, when solved, allows for an explicit calculation of the active stress in the fiber direction. These are the activation stretch-activated ion channels. They have a similar structure to the Hodgkin and Huxley, but they are instead activated by a stretch. This is the stretch, the mechanical stretch. So when there is a certain stretch level, then these channels open and current goes through. So this active contraction model that I mentioned is what I would call the electromechanical coupling. So the electrical problem affects the mechanical problem. And these two here, stretch-activated ion channels, and this modified monodomain equation, it's modified because there is a pullback. There is a pullback of the diffusivity tensors here. What it does is basically solving the problem on the undeformed mesh. This is what I would call mechanoelectric feedback. So the mechanical problem affects the electrical problem, and these terms is what I defined before as bidirectionality. So this is the small electromechanical example that I mentioned. It's basically a small piece of tissue run locally, just 4 cores. It takes a few minutes, and it's a comparison between 2 different excitation contraction coupling models, Hunter here and Land here. LANT is a very recent excitation contraction coupling model. Hello? Okay, yeah. I've been told that there was a problem with the sound, but now it seems to be back. So this is how it contracts. We trigger, I'll run it again. We trigger an electrical pulse in this end, and it propagates, contracting the tissue as the electrical wave passes by. Okay. So Alya has, as I mentioned, 4 different input files. The DAT file, DOM.dat file, CUR.dat file, and EXM.dat file. I'll show it later. So these are 2 of the different input files, the DAT and the DOM.dat. What they define is different parts of the problem. For instance, the DAT file defines the general data of the problem, as the name you want to give to your simulation, the maximum number of steps, the time interval your simulation is considering, and if your simulation is single physics or multi-physics. This $ is a comment, so basically this is not active. If I uncomment this could be a multi-physics simulation, so excitation media, so like electrical propagation and mechanical contraction. The DOM.dat file defines the geometry, the domain of our problem, defines things as the nodes, number of elements, the spatial dimensions, the type of element we're considering, if we have any boundaries that we want to define boundary conditions on, rules of integration. But we also need to include here things as the geometry, the connectivity of the mesh, the coordinates of the nodes, what sort of stimuli we want to consider, and so on. Just before I go on, I just wanted to say that if you have any specific doubt, you want to know something specific, you have the documentation of Alya in this link. And I didn't mention it before, but I was told yesterday, if you want to use Alya, you have 2 options. One of them is through any praised supercomputer, you should be able to use the compiled executable without any problem. Or another option is contacting, I'll give you the email later, Mariano Vázquez from Barcelona Supercomputing Center, and you should be able to get Alya without any problem. These 2 files that I'm showing here are the CUR.dat and the XM.dat. The CUR.dat just defines very few additional data that you need to solve the problems, such as when you want to output and what you want to output. So in this case, we're outputting on the default mesh, and each 50 steps of our simulation, we're gonna dump results on the hard drive. What I said is that if you consider a multi-physics problem, you don't really need to consider coupling, but if you want to consider the coupling between the different physics that you're considering, you need to activate these lines here. For instance, you could be running electrics and mechanics problem, which are not coupled, but they act at the same time. But if you want them to interact with each other, you need to activate this. The XM.dat gives information or needs information about your electrical diffusion problem. You need to state what are the electrical stimuli here. You need to define also the electrical properties of your different materials. You can include different materials, different subdomains within your domain if you want. You need to define the fibers and the cell type. And also, last but not least, you have to define what are the numerical solvers if you want. So you need to define the linear solver in this case, and also the time integration, which is defined here. This safety factor is basically, it has to do with the time step that we are considering. It is saying here that we're considering a time step which is 25 times larger than the critical time step for explicit integration. And just to conclude, I'd just like to mention that in these models we are considering, we're actually modeling quite complex physical and biological phenomena. Obviously, the mathematical techniques are not straightforward. They're not simple, but I mean, with the appropriate code and supercomputing platforms, we are able to do that. The biventricular simulations we're considering are fairly large, a few million nodes and several tens of millions of elements. So we need a supercomputer, basically, unfortunately. Another thing is that our current research is aiming at trying to solve clinical problems and also assessing the effect of drugs, I mean like cardiac drugs, on the heart. With these sort of models, we're sort of aiming at substituting current protocols for completely in silico, so completely computational or mathematical protocols, so that basically we don't put the lives of people or animals in danger. And these tools allow us to explain some of the abnormalities we observe in the ECG, and this is super relevant just in case you want to solve, for instance, the inverse problem. When you get an ECG and you want to know what's actually happening in the heart, this is actually very, very relevant, clinically speaking. And just to conclude, I'd like to just show the people, the acknowledgements, the people we collaborate and that have contributed in this webinar. So thank you. And I've been told that there are not too many questions. So if you have any questions, please write them now, and we'll be collecting them and answering them as soon as we get them, basically. Yeah, so we've received a question about what are the possibilities of defining ischemic regions in the heart for simulations? Okay, so I mean, it is possible with actually both softwares. Anna has showed the possibility in CHASTE, but I can tell you, you can also do the same. You just need to be in Alya. You just need to define another region with those characteristics and just join them together with the healthy regions. But it is possible with both software. Any more questions?
Yes, there's another question from Jenny. Can you see that?
Okay.
I can read it for you. Thank you. Great
Yes.
presentation. Just wondering what kind of testing is done for CHASTE and Alya packages.
Anna will answer about CHASTE. I can answer about Alya if you want. Okay, what routine testing? Yeah, so I mean, in terms of validation, I don't know what exactly testing means here, but I'm just going to ask in terms of validation. I mean, obviously, every time we do a simulation, we compare the results with literature or experiments of our collaborators. So that's not a problem in terms of, as I said, validation. And there is a daily testing check with Alya, basically, if there are some changes that have been uploaded to the trunk and the results change. Obviously, this is highlighted so that basically the problem is solved if there is a mismatch between results. And now I'll let Anna answer about CHASTE.
Hello. Yes, something very similar happens in CHASTE. Everything that they are routinely tested to check that everything is working properly. Indeed, there are like several tests that are run for each modification that is done to the code. So yeah, I mean, nothing more to say. We have received another question that is, have you attempted this modeling in the atria as well as the ventricles, and have you expanded to model arrhythmias? Yes, not myself, but there are people in the group that has modeled the atria and has modeled atrial fibrillation and arrhythmias. So yeah, you can find it. I mean, I can send you, if you send me an email, I can send you some references from the group. People like Carlos Sanchez, Anna Muskevich, they have done this kind of simulations. Yeah, there is another question from John Wormsley, also talking about CHASTE. And in this one is that there is several years ago, there was an implementation of mechanics in CHASTE. Do you know if there are plans to integrate this with electrophysiology at any point? Not by now, but we're open to do it, but we are not working in this line by now. Okay, Frances, do you want to answer?
Which one?
Related to the validation procedure, this is a question of Eduardo Souda. Do you know CARTO software? CARTO software is the clinical tool to study the voltage in the heart. Have you compared your results against CARTO? Yes, we know what CARTO is. We don't personalize all the electrophysiology. It's impossible to personalize all the electrophysiology in the heart because we are different. We have variations from patient to patient, also from region to region. But we use CARTO. We are using CARTO in some of the projects that we are developing in order to feed with that information, that is measures of the electrical activity in the endocardium or epicardium, to insert that information in our models. And like this, we can parameterize our models with patient-specific data. Okay, there is another question about CHASTE. Is the O'Harody model, these softwares use the 211 or modified? Where to get the modified? We are using a modified model, but because the original O'Harody model didn't show the expected electrophysiological changes under ischemic conditions, and that's why we have published the modified O'Harody model, also with the FK-ATP channels. In the paper that I showed, it's data in BMS in this year, 2017. If you ask me for the code, I can send it to you.
Okay, there is Frances back. There is another question saying, is it possible to communicate both codes? What would you get from that? No, it is not possible. I don't think it's going to happen because, in principle, both relevant physics are considered in either of them. So there is no need to do that. There is another question here saying, hi, really nice talk. Are you planning to extend CHASTE Alya to be run in GPUs? I'm not so sure about CHASTE. I think there is no plan to do that in GPUs, but I can tell you there are a few people in Barcelona Supercomputing Center working with GPUs. So as far as I know, the work done with respect to GPUs on Alya is to implement linear solvers, which are GPU based. I think that's everything, but there might be something else. But if you leave your email here, I can send you some information about it. But I'm pretty sure that at least there is work done on linear solvers based on GPU. Linear solving is where like 60, 70, 80% of the time is spent in like big simulations. So that's where the focus needs to be done. I hope I asked your question. I answered your question. So, but as I said, if you leave the email, I'll send you some information about it. Is there any other question? Because I think you answered them all of them.
Yes.
Oh, yes.
And then
yeah.
Let me see how to answer that, if we can write or something. Yes. Okay. What we'll do instead is we'll put our emails. So if you need anything, just email us. Okay. If you have any further questions, please just send us an email. It should be fine. Yes. And if you want any information regarding GPUs or regarding the atrial work, just send us an email, please.
Okay, great.
Okay, so I think that's it then.
Yeah, shall I close the webinar then? Thank you very much for presenting and
thank you. Thanks to everyone for registering.
Yeah, and all the material of the webinar you can find on the CompBioMed website and the VPH Institute website. So if you want to see it back, you can find the recording over there. And please fill in a small feedback questionnaire that will appear if you close this webinar panel. Yeah, thank you, and hope to see you on the next series of the CompBioMed webinars. Bye.